Welcome back to Dr. Sears biology class. This is our Meiosis Project by Jonathan Wang and Fredrica Peng. I'm sure as this chapter started, you've all heard the different terms zygotes and gametes, mitosis and meiosis, chromosomes, chromatids, and chromatins, and we're confused by the similarities. While one of the hardest concepts to learn is meiosis, due to its similarity to mitosis, another form of cell division. So, what exactly is meiosis? Starting off, we have interphase, which is comprised of three main steps: G1, S, and G2 phases, and two checkpoints, G0 and G2 checkpoints. Interphase is mainly when the cell rests, where it spends 90% of the cell cycle preparing for the next time it does meiosis. Now, this is the first step, G1, or gap 1 phase. The cell acts normal here. It has two chromosomes N and one chromatid per set, X. They have all the DNA packed into a purple nucleus. In this phase, the cell grows as well as synthesizes materials for later. However, what comes next is actually a checkpoint. This checkpoint is called the all or nothing checkpoint, and it is called that for a good reason. If a cell goes through this checkpoint, they must continue the cell cycle. However, if the cell does not go through this checkpoint, they must go into G0 phase, in which they can no longer grow. After the G0 checkpoint is the synthesis phase, aka S phase. In this stage, you guessed it, stuff is synthesized. What that stuff is the cell's DNA. They do this by copying chromatids, creating sister chromatids per one chromosome 2N, 2X. The next phase, gap 2 phase, is all about prep work. This phase is right before the M phases, so it must store food, synthesize proteins, as well as copying the organelles needed for M phases. One last check before takeoff. It's the G2 checkpoint. Here, a cell is checked for DNA damage. If it has DNA damage, then it will try to fix the damage or make it commit apoptosis, cell death. If the cell is fine, the cell proceeds into M phase. Now, after all the interphase is the official start of the meiosis 1 phase. M1 phase consists of the same 5 parts of mitosis: prophase 1, metaphase 1, anaphase 1, telophase 1, all combined to make PMAT, and cytokinesis 1. This is prophase 1, and quite a bit happens. First, the yellow centrosomes consisting of centrioles and spindle fibers duplicate and start moving to the poles. The nuclear membrane also starts to dissolve. Then the DNA in that nucleus compacts in the chromosomes. The chromosomes combine through synapses, forming tetrads. When they combine, chiasmas form, which allows them to cross over DNA. Metaphase 1 is next, and it is much simpler. The centrosomes are at the poles and the nuclear membrane has dissolved. The tetrads then line up in the middle, as you can see here. Then, the spindle apparatus from the two centrosomes connect to one another. The ones at the center are called the kinetochore microtubules. Anaphase 1 is simple enough. The homologous paired chromosomes are then ripped in half, as the kinetochore microtubules pull from each other, and the non-kinetochore microtubules push each other, elongating the cell. Then comes telophase 1. In telophase 1, the chromosomes reach the poles of the cell, where they begin to unravel, while a nuclear membrane forms around them. As the cell elongates, the cell membrane starts to pinch off, forming a cleavage furrow. The last step of M1 phase is cytokinesis 1. Here, the two cell membranes finally separate, each a haploid cell, different from the parent cell. Once the first round of PMAT is finished, there's only one more needed to complete meiosis, and it is called meiosis 2, or M2 phase. Unlike before, the cells from M1 do not need to go through interphase, but rather go straight into M2 phase. For prophase 2, the process is around the same as before, except everything happens twice now, one per daughter cell. The centromeres duplicate, the nuclear membrane disintegrates, and the spindle apparatus starts to form. The same applies here to metaphase 2. In this phase, the chromosomes line up single file on the metaphase plate with a non-specific order. This is because when the tetrads were ripped apart, what resulted were many individual chromosomes independent of each other. In anaphase 2, nothing new happens here as well. The sister chromatids begin to separate and move to the poles, and the non-kinetochore microtubules elongate the cell. Well, what a surprise! Telophase 2 happens to be around the same as the previous telophase. In telophase 2, the new nuclear membranes form, the chromosomes uncoil, and the cell membrane tightens for cytokinesis. And finally, for the last step, cytokinesis 2, it unsurprisingly acts as before, with the division of the cytoplasm. However, this time, it has the creation of 4 new unique haploid cells. So now, after all that, what was the point? Why did we have to go through all that twice instead of just going through meiosis, which only does it once? Well, for starters, meiosis is important for the creation of gametes, which are human sex cells. In females, this process is called oogenesis, in which meiosis creates one egg cell and three polar bodies. This means that each cell has the same DNA in it, but the egg cell has a larger amount of cytoplasm than the other three, causing only it to develop into an ovum. In males, meiosis creates from one cell 4 equal sperm cells, all equal in DNA content and size. However, its most important function lies within you. There is no one out there who is exactly like you. Meiosis makes sure that every individual is unique, which can be seen in prophase 1, when the chromosomes swap their DNA, causing variety. So thank meiosis for who you are, and thank you for watching our presentation.